Dear students, our next topic is the polarizable and the non-polarizable base electrodes. First of all, electrodes are employed to pick up the electrical signals of the body. That is, to presenting in a bioelectric event to the pre-amplifier of signal processing circuit, a pair of electrode plays the role of a transducer. That is, electrodes are transferring the bioelectric event to the input to the amplifier. The amplifier that accommodates the characteristics of the electrodes. And these types of electrode to be used depends upon the anatomical location of bioelectric event and the dimensions of the bioelectric generator. And in this theory we have discussed about the two types of electrodes, that is polarizable electrodes and the non-polarizable electrodes. In case of a polarizable electrodes, no charge crosses the electrode-electrode interface when a current is applied. That is, when a current is applied, no charge is formed between the electrode-electrode interface. For example, a platinum electrode. In case of a non-polarizable electrode, the current passes freely across the electrode-electrode interface. That is Ag and AgCl electrode. That is the two types of polarizable and non-polarizable electrode. Second is the equivalent circuit for biopotential with the two electrode. That is, we have to see the two electrodes, electrode A and electrode B, and the cell resistance and the cell potential, and we have to take output from the right side. That is all about the equivalent circuit for a biopotential with the two electrode. Next is the biopotential electrodes. As you will know, to pick up the electrical signals of the body. That is, these biopotential electrodes transduce ionic condition to electronic condition. That is, it acts as a transducer to convert the energy from one form to another so that biopotential signals can be obtained. They generally consist of metal contact packaged so they can be easily attached to the skin or the body tissue. So that we have to take the signals from the body tissue or body parts from the action potentials. Then is the classification of electrodes. The electrodes, there are three types of electrodes. First one is the microelectrode, and they are used to measure the bioelectric potential near or within a single cell, and these are also called as intracellular electrode. So just note the point, the microelectrodes are also known as the intracellular electrode, that is used to bioelectric potential from a single cell, and its tip must be tungsten or stainless steel. Next is a micropipette. The micropipette with the size of 1 micron. It is made of glass. It is filled with the electrolyte. Then is the skin surface electrode. This skin surface electrode we have to measure about ECG, EEG and EMG. And these are used to measure the potentials available from the surface of skin and are used to sense the potentials from heart, brain and nerve. So we measure ECG, EEG and EMG. Next is a needle electrode. Needle electrode also known as depth and needle electrode. And these are used to measure the bioelectric potentials of the highly localized extracellular regions in brain or bioelectric potentials from a specific group of muscles. That is, we have to that is it will penetrate the skin to record the EEG. So we have to take the extracellular regions in the brain or the bioelectric potential from the group of muscles. That is called a skin surface electrode or depth and needle electrode or the microelectrode. Then is the microelectrode. The microelectrodes are mainly divided into metallic and non-metallic. The non-metallic microelectrode is called micropipette. The microelectrodes should have smaller diameter and during insertion of the electrode into cell, and there will not be any damage to the cells. In this theory, to measure potential across cell membrane, that is, it is very smaller in size with respect to the cell dimension. It is very smaller in size, and the tip diameter ranging from the 0.05 to 10 micrometer, and it does not change the cell's behavior, and it's formed from a solid metal needles or a metal contained within or a surface of a glass needle. and a glass micropipette having a lumen filled with an electrolytic solution. And I already told you the two types, metal and micropipette. First of all, the metal electrode, and in case of a metal electrode, they are formed by electrolytically etching the tip of a fine tungsten or stainless steel wire to a fine point. So the fine needle of a strong metal, and it is made of stainless steel or a platinum iridium alloy, and made of tungsten or a compound tungsten carbide. Here is the figure of a metal electrode. In this case, this technique is also known as electropointing. That is, the metal microelectrodes are coated almost to the microtip with an insulating material. That is, in the figure we can see that a insulation from the side to the tip of it. To reduce the impedance, some electrolytic processing like chloriding the tip, that is usually produced by electrolytic etching using an electrochemical cell in which the metal needle is the anode. That is, to reduce the impedance, that we have to make some chloride to chloride the tip, and then developing by the photographic developer can be performed. The electric current etches the needle as it is slowly withdrawn from the electrolytic solution. The etched metal needle is then supported in a large metallic shaft that is then insulated. That is the main working behind the metal electrode. And next is the micropipette electrode.